Dear students, welcome back to Biology Forum classes. This is the only platform where you will get concise, concept-based and information-rich videos on life science. Starting from elementary level up to graduation and post-graduation level, as well as we also cater students from competitive examination as well. Okay? So, before starting today's class, let's quickly recall our previous class in which we have studied about ethylene hormone, the definition of ethylene hormone, the discovery and bioassay, precursor molecule, its site of production or site of synthesis you can say, various example, and the most important is the physiological role of ethylene hormone, mainly present in two forms. The one is as ethylene itself in the gaseous form, and the second one is in liquid format, which is named as ethapon and vigorously used in agricultural purposes. So this is the concise recall of previous class. Today we will about to study abscisic acid, which is the most important growth inhibiting hormone in case of plants. So without further ado, let's begin today's class. Okay. So to study about abscisic acid, we can say from the name itself that the nature of the particular hormone is acidic. The hormone is acidic in nature, and generally we can say it as a general growth inhibiting hormone. Because other four hormones somewhere act as growth promoting or cell division promoting, but this is the only hormone which act as growth inhibitor hormone in case of plants. So quickly study about its discovery. To study about the discovery, mainly the hormone was discovered somewhere in 1960s, in the era of 1960s. Okay, a group of scientists, there is a background story in case of this particular hormone. What is the background story? The background story is somehow like that. A group of scientists have examined or going through an experiment on certain plants, we can say, and they have found that there are some substances, some chemical substances or some molecules are there in the plant itself which are produced in the plant itself, which act as a growth inhibitor or growth inhibiting particle. Okay, so they categorize those particles in three categories. So what are the three categories? The first is inhibitor beta. Okay, the first is inhibitor beta. What is the second one? The second one is abscission 2, Roman 2, abscission 2, and the third one is dormine. Okay, the third one is dormine. Okay, so a group of scientists during their investigation found certain type of chemical molecules which act as growth inhibitor particles in case of plants, and they categorize them into these three categories. The first one is inhibitor beta, which they found in which plant? In case of potato tuber. In case of potato tuber. In case of potato tuber, they found the first inhibiting particle which they name as inhibitor beta. Okay. The second one is abscission 2. Okay, the second one is abscission 2, which they found in fruits of cotton plants. Fruits of cotton plant. Okay, fruits of cotton plant. And the third one is dormine. Okay, the dormine they found in the leaves of betula plant. Where? Leaves of betula plant. Okay, I am just removing this book from here. Okay, so these are the 3 inhibitors. The first one is inhibitor beta, which they found in potato tuber. The second one is abscission 2, which they found in fruits of cotton plant. And the last one is dormine, which they found in the leaves of betula plant. But after vigorous investigation, they have found, or they have concluded that, or they have made a conclusion that all these 3 act as growth inhibitor. That's why they grouped these 3 molecules, and together the 3 molecules they name as ABA, or abscisic acid. ABA is the short form of abscisic acid hormone. As these 3 individually acting as growth inhibitor particles, together they name these 3 as abscisic acid hormone, which is the first growth inhibiting hormone in case of plants, we can say. Okay, so far about up to this is the discovery part, how this hormone was discovered and in the 1960s. Okay, so in the next part we will study about the bioassay site of synthesis part. Okay, so let's continue. Okay, so coming to the next coming to the next part is the bioassay part. After the discovery, the next part is the bioassay. The bioassay part is not so important for you students. It is not so important in your syllabus, and also it is not mentioned in your syllabus, but still I am mentioning it over here that the test for bioassay in case of abscisic acid hormone is the anti-transparent test. Just remember this. If there is any question related to this appear in your exam, just remember up this point is that the bioassay experiment is named as anti-transparent test. What is anti-transparent and how it is related to abscisic acid hormone, we will study later in when we will study about the physiological role. Okay, coming to the next part, the precursor molecule. In our previous lectures we have found, or we have already studied, that there is one precursor molecule definitely present for each of the hormone, from that particular precursor molecule the hormone will get produced. Okay, here is nothing different from the previous one. Here is also there is a precursor molecule which is carotenoid. So we know that carotenoid is mainly responsible for different colors in plants, different colors of fruits, colors of leaves, colors of flowers. Okay, so from carotenoid specifically, if we go into depth, so we can say that here the precursor molecule carotenoid is viola xanthine. This is the precursor molecule from which after oxidation, after getting oxidized, it will produce abscisic acid. After what? After oxidation, the hormone abscisic acid will be produced from which? From viola xanthine, which is one kind of carotenoid. Okay, already present inside the plant. Okay, so next coming to the site of synthesis or site of production of abscisic acid hormone. Okay, so there is three sites. There are 3 sites of production. First one is the dormant seeds, second one is the site of senescence, and third one is the site of abscission. Okay, so quickly discuss about this. Dormant seeds. Dormant seeds are those seeds which will not germinate, the seeds which will remain in hibernated or sleeping situations. They sleep. Okay, just remember that whenever you get the term dormant, the dormant term means that the seeds will not get germinated, or the seeds are in hibernation or sleeping position. There is no activity going over there. Okay, so this is the first site of production of abscisic acid hormone. The next is at the site of senescence. We know from the previous lectures that senescence means aging. When a leaf or when a fruit get ripened, or when a leaf will attain an age of maturity, it will fall. It will detach from the petiole, or it will detach from the stem, and it will droop down. Okay, so at that particular site, the concentration of abscisic acid hormone is much higher than the other portion of the plant. Okay, so the second part is at the site of senescence. The hormone will get produced. The last is at the site of abscission. So senescence and abscission is interrelated. Like senescence and how? After getting mature, any fruit or any leaf will get detached from the plant, and the detachment is termed as abscission. Okay, so you can just think how beautiful the nature is working on itself, that after in case of human also, in case of animals also, after attaining a certain age of maturity, an animal or a human being like us, we will die. Okay, after a particular certain point of time, after a lifespan we have already covered, we need, every living thing need to die. In case of plants, as plant is also a living entity in our world, it, when its leaves or when its fruits attain the age of maturity or get ripened, it will definitely detach from the petiole or from the stem, and it will droop down or it will fall, and this is called abscission. And at the site of abscission, the hormone, which is already named as abscisic acid, the concentration is much higher in that particular site. So the site of synthesis or site of production, we can say, are three. The first one is dormant seeds, the second one is senescence, at the site of senescence, and the third one is at the site of what? At the site of abscission. Okay, I think up to this you have understood well. Okay, so let's continue the last portion of our class, which is the physiological role of abscisic acid hormone. Okay, so let's continue. Okay, so to study about the physiological role or function of abscisic acid hormone, the first one is abscission. From the name again we can say, as the name of the hormone is abscisic acid, it must have a function over abscission. After over detachment of fruits or leaves from the plants, the hormone has a very high role. Okay, the second one is the hormone inhibit seed germination. Okay, to study about seed germination, from the lecture 2 about gibberellic acid or gibberellin, I think you can recall the fact that gibberellin hormone enhance the seed germination procedure. So quickly I make you recall about the seed germination procedure. So this is a growing seed. This part is what? The embryo, which is inside the seed. Okay, let's use a different color. This is the endosperm portion. Just I am giving a tip over here, or an additional information, that in case of monocot plants like rice, the portion of rice which we eat actually as our daily staple food is the endosperm portion, which is rich in starch. Okay, this is just a small information for you guys. Okay, so this is the embryo, this is the endosperm portion, and outside the endosperm portion, is a layer which is named as alluron layer. Okay, so this is the structure of monocot seed. Of what? Monocot seed, like rice you can say. Okay, this is the embryo over here. This is the black portion is the endosperm, which is rich in starch, which is a complex form of carbohydrate, and the red layer I have drawn here is the alluron layer, which is surrounding the endosperm portion. Okay, now the interesting thing is that in case of the alluron layer is rich in a particular type of enzyme. Enzymes are certain biochemical molecules which either accelerate or decrease the level of any accelerate or decrease the speed of any chemical reaction. This is the enzyme we can call. Okay, so in the alluron layer, what is the name of the enzyme? The name of the enzyme is amylase. I think you can recall from the lecture 2, right? Okay, amylase is carbohydrate solubilizing enzyme, or the enzyme which can break carbohydrate. Okay, so I have just mentioned that the endosperm portion is rich in what? Starch. Okay, starch is a complex form of carbohydrate. This amylase enzyme will break the starch into maltose, which is another form of carbohydrate, and at the last part the maltose will get converted into glucose. What? Glucose, which is the simplest form of carbohydrate. We can say monocarbohydrate. Okay, and the glucose is the rich source of nutrition for the developing embryo over here. The blue one is the developing embryo, and in case of a seed, there is an embryo, and the embryo need to have a source of nutrition for its development, and after getting enough nutrition, the seed will get germinate or will germinate eventually. Okay, for germination, I make it clear for you, not only the nutrition but several other factors are also responsible, like the humidity, the temperature, the natural calamities. Okay, mainly the natural parameters. So come to this point, amylase enzyme which is present in the alluron layer will break the complex carbohydrate or starch present in the endosperm into maltose. Okay, from maltose the enzyme maltase will convert it into glucose, the simplest form of carbohydrate, which is the primary source of nutrition for the developing embryo over here. Okay, and what is the function of gibberellic acid here? The gibberellic acid will enhance the effect of amylase enzyme. Gibberellic acid or gibberellin will enhance or will increase the function or the activity of amylase enzyme, which is present already there in the alluron layer. Okay, so this is the case of seed germination. Okay, this is the process how seeds are getting germinated. Okay, so how it is related to, and how I am saying over here this, already I have mentioned this in my previous lecture. The relation is that the seeds are in the dormant state due to the presence of excessive amount of abscisic acid hormone. In case of you can relate to in day-to-day life that the seeds, when it is not the season of any vegetable or any fruit, the seeds we can if you can recall that the term cryopreservation or seed bank. Okay, in the seed bank the seeds of various plants are preserved in a particular manner. Okay, or in the simplest form, just you can conceptualize it like this. We have got a handful of seed of any plant and we put it in a bottle and just tightly put the lid on and keep it like this. The seeds will not germinate. The seeds will remain in the dormant state in that particular plastic or glass bottle. Why? Because in case of dormant seeds, or in case of those seeds which we kept in the confined environment, the amount of abscisic acid hormone, which is a growth inhibiting hormone which inhibits seed germination, the amount or the concentration of abscisic acid hormone in that particular seeds which are kept in a confined situation is so much higher that it overcome the effect of gibberellin hormone, which mainly target the seed germination. Understood? I just want to mention here that if the level of ABA, abscisic acid, is greater than gibberellin hormone in seeds, then the seeds will not germinate. Okay? Because the abscisic acid, I have just shared, inhibits seed germination. But if the situation is favorable, and if there is some kind of environmental favorable situation going over there, then if the gibberellin hormone concentration is higher than abscisic acid hormone, then the seeds will definitely get germinated, because the gibberellin hormone is the sole cause for seed germination. Okay? Just imagine how beautiful the subject is and how beautiful our nature, mother nature is, that they control the concentration of that particular hormones in such a beautiful manner that whenever we need that the seeds will remain in dormant state, it will. If we apply artificially synthesized abscisic hormone from outside, the seed will not get germinated, it will remain in the dormant state always. Okay? But if the concentration of gibberellin hormone is greater than the abscisic acid hormone in nature, in case of naturally in plant, then the seeds will definitely get germinated. So this is the second function of, or second physiological role of abscisic acid hormone, is that it inhibits seed germination. Okay? Just related to this, the third function is it maintains the dormancy of buds and seeds. Maintain dormancy in buds and seeds. Just I have mentioned the second point is it inhibits seed germination. Inhibition of seed germination means what? Means the seeds or the buds will remain in sleeping state or hibernated state or dormant state. Okay. So the maintaining dormancy in buds and seeds is the third important function of abscisic acid hormone. Okay. Okay. So I think I can erase the board and let's come to the fourth function. Students, please note as the class is going on. All these points are very important for your upcoming examinations, in case of medical, in case of NEET, or in case of board examination as well. Okay. The fourth function of abscisic acid hormone is that it inhibits protein synthesis. How it will work? Okay, it will work in a particular manner. What is protein synthesis? At first we have to know this. Protein synthesis, protein mainly produced from RNA, ribonucleic acid. Okay, and from where RNA are formed? RNA are formed from DNA, deoxyribonucleic acid, by the process of transcription. So DNA to RNA is the process transcription. In our molecular biology and cell biology class, we will study about this. And from RNA, protein will be produced by the process of what? Translation. This is not the translation of English, okay? Just joking. Okay, from RNA to protein, the process of production of protein from RNA is termed as translation. From RNA, several amino acids which are there in our body as in the amino acid pool, one by one amino acid our body will pick and it make a polypeptide chain, and a total polypeptide chain will be act as a protein molecule. Okay, so abscisic acid hormone does what? As it is a growth inhibiting hormone, it will inhibit the process of translation. As a result, it will inhibit the protein synthesis process. Okay, if there is no enough protein in the environment or in the plant body, actually we know that protein, carbohydrate and fat or lipid are the building blocks of our body, in case of human or in case of plants also. So if there is not enough amount of protein present in the body, several other functions will get turned off. Like how? Suppose this is a plant, this is a stem, and this is a petiole, and sorry, my drawing is horrible. Okay, so please bear with me. Okay, I think you can have understood, you can understand. Just a rough drawing over here. Okay, this is the growing leaf, this is the growing leaf, the stick is termed as petiole, and this is the stem. Okay, if there is not enough amount of protein in the body, as abscisic acid concentration if higher, it will disrupt the process of translation and no protein synthesis will occur. Then as a result, the leaf will attain an early age of senescence. It will get mature very early. Okay, it will senescence. It will face senescence, very early senescence. I think you can see the board clearly. If not, just let me overdo this. Early senescence. As a result of lower concentration of protein in the body, the leaves will attain an early senescence, or it will face early aging and it will droop down eventually. Okay, it will cut from here. The stem will not get enough protein supply, the petiole will not get enough protein supply, and as a result the plant will die. After a certain period, the plant will die. Okay, so this is the drawback of abscisic acid hormone. The function is that it inhibits protein synthesis, and as a result the plant will die and it will attain an early age of maturity or early senescence. It will face. Okay, so this is the fourth function. The fifth one is the fifth function is, I think you can recall, I have mentioned in the early class today itself, is that the bioassay's anti-transparent test, and I have also mentioned that we will discuss this anti-transparent process in case of physiological role. Okay, so the time has come. The hormone abscisic acid is also named as anti-transparent hormone. How and why the name is given? We all know transpiration means water loss. Okay, in case of human or in case of animal, during summer season, vasodilation occur. What is vasodilation? The arteries and the veins are dilated. Why? Because there is enough humidity or enough temperature in the environment also already there, and by vasodilation our inner body temperature will decrease by excreting sweat. We excrete our toxic materials which are produced eventually in our body, and we will sweat and release all the toxic materials in the form of sweat from our body, and this is mainly caused due to the process of vasodilation. Just like this, plants also have a similar procedure over there. Plants face actually plants do the process is called transpiration, by which by transpiration means they release or excrete water by stomata already present in the leaf. If this is a leaf, it is not prominent, I am right. If this is a leaf, there are several pores are there which are named as stomatal opening in the leaves, okay, and by these pores they excrete water and all the metabolic byproducts produced in their body, okay. So this abscisic acid hormone will cause closing of stomata. Okay, how or why does it function like this? If a plant face a drought situation, no water supply, no enough water supply will be there, or lower amount of rainfall will be there, or in desert condition, if they face the process transpiration, there is no enough water in the environment, and side by side they will lose water from inside of their body through transpiration. So as a result, if these two processes are going simultaneously, after certain point of time the plant will die. So to prevent the plant to get die, the abscisic acid come in action. What kind of action? They close the stomata, the pores present in the leaves in the plant body, which causes water excretion from the plants, okay. They close the pores, they close the stomata, and as a result water is conserved or preserved within the plant body. If there is not enough amount of water present in the outside region, or if the plant will not get enough water supply, if there is any drought condition going on, if there is lower amount of rainfall, or if the plant is in desert condition, this hormone will close the stomata, and as a result water will be preserved or conserved within the plant itself, okay. So that is why the abscisic acid hormone is named as anti-transpirant hormone, okay. Coming to the sixth point, this hormone helps in stress condition. There are so much stress condition is going on in our environment. Low rainfall, drought, for say nowadays it is we everyone, not only the plants, we also face these environmental changes very vigorously, say global warming or rising temperature, okay, rising temperature. All these unfavorable conditions, this abscisic acid hormone come in action. They help the plant to sustain in stress condition, to maintain itself in stress condition by closing the stomata and by letting it preserve the water already present within their body, so that if there is low rainfall, if there is drought, or if there is global warming, or if there is any type of unfavorable condition is going over there over outside, the plant will remain or maintain its sustainability in a particular way, okay. So I think up to this point I am clear to you, and you have understood well the physiological role of abscisic acid hormone, okay. So here is another information I would like to add over here. This is very important for your NEET examination, is that every time many times the question will appear that what is the antagonistic hormone of abscisic acid. Abscisic acid is a hormone in case of plants. And what is the antagonistic hormone already present there in the plant? Okay. So the answer is gibberellin or gibberellic acid. Gibberellic acid and abscisic acid will have an antagonistic relationship. How? In case of seed germination, we have just studied that if the level of gibberellin is higher and abscisic acid is lower, then the seed will get germinate. And if the scenario is just opposite, the level of abscisic acid is higher and the level of gibberellin is lower, then the seed will remain in a dormant or sleeping or hibernating state. Okay, that is why we say that the abscisic acid as anti-GA, or what we say anti-GA or antagonistic hormone to gibberellic acid or gibberellin. Okay, up to this point I think I have make you understand about the physiological various roles of abscisic acid. Okay, so up to this I think the total part of the 5 important plant hormones, auxin, gibberellic acid, cytokinin, ethylene, and the 5th one and the last one we have studied today is the abscisic acid. Almost the syllabus is covered. Okay. Okay. So after completion of total 5 hormone in case of plants, an additional information I would like to add here is that synergistic action and antagonistic action of hormone. Okay, I have already written it on the board. Just continue. In case of synergistic, just complete the left side first, then we will go to the right hand side. Okay, here synergistic effect or synergistic action means when two hormones act in a similar manner. When two hormones will promote something equal or something similar. Like what? For example, the first point, promotion of cell division. Can you tell me the name of 2 hormones which will promote cell division? You can recall. Can you recall it from the previous class? Okay. Promotion of cell division. 2 hormones act over this. What are the 2 hormones? The first one is auxin. Okay. And the second one is cytokinin. The first one is auxin. I think now you can recall this, right? Auxin and cytokinin both are the 2 hormones which promote cell division. Auxin causes cell elongation and cytokinin causes cell division. Okay? Because the cytokinin, the name comes from that the hormone cytokinin helps in cytokinesis, that is the division of cytoplasm. What are the next part or other parts of cell division? Karyokinesis, which is the division of nucleus. Okay, division of chromosome actually. So auxin and cytokinin are 2 hormones which act as a synergistic action to promote the cell division. Okay, the first part we have covered. The second part is abscission, detachment. Abscission, detachment. What are the 2 hormones that function in a synergistic way for abscission? The name is abscisic acid and ethylene. Ethylene and ABA. How? Ethylene is commonly named as fruit ripening hormone. When a fruit gets ripened, it will droop down from the tree. When a leaf attains an age of maturity, it will detach from the tree, and all these phenomena will take care by ethylene hormone and also abscisic acid hormone. That is why these 2 hormones function in a synergistic way for abscission part. Okay, up to this I think I am clear enough. Let's come to the right portion. What is antagonistic? Antagonistic, I have just mentioned, is that when 2 hormones act opposite to each other. Okay, not synergistic way, just opposite. The first one is seed germination. What are the name of the two antagonistic hormone is abscisic acid hormone and gibberellin. Gibberellin and abscisic acid hormone. The gibberellin will promote the seed germination and the abscisic acid will inhibit the seed germination. That is why they act in an antagonistic manner. And that is why the abscisic acid hormone is named also as anti-GA, anti-gibberellin. Okay. The second point is apical dominance. What is apical dominance? From the previous class, I think you all can recall, students. Apical dominance is the formation of apical bud at the top of any tree, and that is why due to the apical dominance, all the lateral buds will not form. And what are the two hormones that act in an antagonistic in particular this case? The name of the hormone will be auxin and cytokinin. I think you all can recall. Auxin will promote the apical dominance. In the presence of enough concentration of auxin, only the apical bud will form, not the lateral bud. But if there is cytokinin hormone concentration is much higher than the auxin, then there will be no apical dominance seen. Only the lateral buds will form. Okay. So in this particular manner, in case of apical dominance, the auxin and cytokinin will act in an antagonistic way. Okay. And the last part is senescence. What are the two hormones here? ABA or abscisic acid and auxin. Abscisic acid hormone, from the name itself we can say it will promote senescence, and auxin will inhibit senescence. That is why in case of senescence, abscisic acid and auxin will act in an antagonistic manner. Okay. I think up to this you have understood well, and this is very, very important. I think you should write it down, the table, write it down in your copy. This is very much important for your upcoming examination, and I will also attach additional group of summary at the end of the episode, at the end of today's class, for your better information. Okay. Okay, students. So in today's class we have completed the hormone or phytohormone portion, main 5 hormones, which are auxin, gibberellic acid, cytokinin, ethylene, the fruit ripening hormone, and abscisic acid. Again I am repeating: auxin, gibberellic acid, cytokinin, ethylene, and abscisic acid. The 5 most important phytohormones which are present in the plants. We have covered the total syllabus according to NEET, according to NCERT, you can say everything. Okay. If there is any question from your side, you can comment down below, or you can mail me in my email ID, which is right now popping in your screen. If there is any doubt or any query, anytime you can contact me via my email ID. Okay. In the next class, we will study about the Vernalin Florigen in very compact manner, and we will start to study about seed germination procedure. Okay. And as the class will proceed, I think you can make yourself comfortable with the learning process I am following. And if you like the video and if you enjoy the learning process with me, don't forget to share the video as much as possible, and subscribe to my channel and put the notification bell on always, so that every time I upload a video, you will be get notified. Thank you so much.